So I'm going to end talking about stem cells, what we know about it. We have educated you about a variety of things from genetics to immunology to neuroanatomy. Well, I'm going to give you a little bit about stem cells, and then I'm going to tell you how we're applying this. And I'll give you the punchline right now. And the punchline is that we are progressing forward with stem cell preclinical studies to the point where we hope to begin enrolling patients in our first stem cell clinical trial next year. Now, there are some variables that will potentially affect this, but we are on course to gather all of the necessary preclinical data, which means the data required by the FDA to justify a human clinical trial by late 2008. And if that goes well, and we don't know for sure if it will, but if that preclinical data looks good, then we will apply to the FDA for permission to begin our first trials in patients next year. There's going to be 2 trials. One is going to be in patients with transverse myelitis, and the other one will be in babies with a fatal paralytic disorder called spinal muscular atrophy. Now, before we begin the trial, the FDA has to grant us permission. So we've been discussing with the FDA, but if all goes well, we hope to begin in 2009. So, Chris, can we get my first slide up? Or do I have it? There you go. Okay. So let me give you then a discussion about stem cells, what we think about, what are some of the definitions. This is all the stuff that you're going to hear as we get closer and closer to that clinical trial. And so there are some definitions and some things that you should probably know about this. I should start right up front and just tell you some of my close collaborators. You all know about Adam and Chitra, but specifically with the stem cells, there's a very good group at Johns Hopkins, and Ben has been critical in advancing these forward. I have several other collaborators outside of Hopkins. Mahendra Rao is at Invitrogen, Steve Goldman is at Rochester in New York, and Hans Kierstead is a really good stem cell biologist at UC Irvine. And we really work as a team, sharing data, sending stuff to each other. And so I think because of that collaborative spirit, this stuff has moved forward more rapidly than it would had any of us said, no, I have to keep this to myself and not share. So that's a real testament, I think, to a good academic environment that allows this to be done. So let's do these definitions. So a stem cell, you hear about all the time. A stem cell is a cell that has the potential to become a specialized cell, but also can maintain its own identity as a stem cell. So it's called an asymmetric division. And what that means is that every time a stem cell divides, it can give rise to a cell that becomes a mature liver cell or nerve cell, but it also maintains its own identity so that it is essentially immortal, right? Because you always have, after every division, the retention of a stem cell, okay? So that means that when we talk about a true stem cell, it can live forever. And you can continually keep that in a stem cell state, and then you can bias certain differentiations toward particular cell types. If I'm interested in liver disease, I'll push them towards hepatocytes or neurons or gulls, etc. Now, a progenitor cell is not a true stem cell because it cannot maintain itself as a stem cell. It's not immortal. It has a finite number of things that it can differentiate into, and it does not maintain its stem cell state. So we're going to talk about progenitor cells because in some respects they are easier to work with. A stem cell is both very powerful, but in some cases a little bit scary. And so some of the earlier trials that we will talk about will use progenitors because they're already committed to becoming something, and you really can't change that. And in some cases, that's quite comforting. So where can we get stem cells, or indeed, where can we get progenitors? You can get them anywhere from a very early stage embryo an in vitro fertilization, and that's, for example, embryonic stem cells. Now, the name embryonic stem cell, that's a terrible name. If we had just named it differently, we would have never had the trouble, because the name embryonic stem cell makes you think of an embryo, right? With tissues and organs and fingers and a beating heart and things like that. And that's where all this controversy comes from. I'll show you where an embryonic stem cell comes from. This little cluster of cells there, smaller than a pinhead, no tissues, no organs, none of that stuff that most of us, I think, would say means really life. Okay? So that's an embryonic stem cell. It comes from the dish, from an in vitro fertilization, from a small cluster of cells that are totipotent, meaning that they can become any cell type in the body. Now, as we progress down further along human development into fetal life, there are fetal stem cells that can come from the fetus. There are also stem cells that can come from the cord blood taken at the time of the delivery of a baby. There are placental stem cells. There are adult stem cells. And there are even tumor stem cells called embryonal carcinoma cells. So at every stage in human existence, there are stem cells. We have stem cells in every tissue in our body. So if we were to do a brain biopsy of any one of us, you could culture out stem cells, and you could bias them toward becoming a certain cell type, and you could even imagine using them for a particular therapy. Now that's new. All of that stuff, this idea that we've all got stem cells in our body, in our tissues, that are attempting to repair disease on a regular basis, and certainly after injury, is all new within the last 5 years. Now, what we don't yet know is why they fail under many circumstances, and how can we harness them to do better, and we're getting there. So we're starting to figure out how to accelerate their ability to repair tissues, but we're not there just yet. So this, again, is the embryonic stem cell. Sperm meets oocyte in the dish for a couple who can't get pregnant the regular way. So in the dish now, you get a couple cells here, and ultimately you get what's called the inner cell mass, and it is this cluster of cells. These should be called blastocyst cells, not embryonic stem cells. And we would have never had this problem, because nobody cares about a blastocyst. It's just a cluster of cells here. But those cells can be taken from this blastocyst and are immortal. They can be grown forever. Some of the cells that were derived back in the 1960s are still growing as embryonic stem cells now. One very good embryonic stem cell line could potentially treat 100,000 people. But yet we've still got all this problem and all this controversy about the politics of embryonic stem cells. Now, in some cases you need embryonic stem cells. In other cases, adult stem cells may be better. So, for example, embryonic stem cells are pluripotent, meaning they can become anything. Adult stem cells are usually much more limited. They can only become the tissues from which they were isolated. So a liver stem cell, kind of makes sense, is going to become a liver cell very well, might become a bone cell a little bit, or a blood cell, but it's not going to become a neuron very well, right? So it's a much more limited potential. The embryonic stem cells are highly proliferative. They divide like crazy, whereas adult stem cells are much slower to divide. Embryonic stem cells can be used in a non-autologous way. In other words, if we're developing motor neurons from an embryonic stem cell, that will be transplanted into somebody who is not genetically identical. But yet adult stem cells, you can imagine them actually being transplanted back into the patient from whom they were isolated. So you can isolate from a particular patient a stem cell, culture them, do what you want with them, and put them potentially back into that own individual patient. So there are ethical concerns about embryonic stem cells. One of the things that we worry about is, are they tumorigenic? Can they form tumors? sometimes the fact that they are so powerful worries you, because they can become anything. But what if they become a tumor or cartilage within the spinal cord, for example? That's a bit worrisome. So, for example, some of you may have been following Neuron in their attempts to have an embryonic stem cell-derived clinical trial for patients with traumatic spinal cord injury. And Neuron had been doing for the last decade a very good job of developing these embryonic stem cell-derived oligodendrocyte precursors. And we're progressing toward a clinical trial. I won't get into what the clinical trial was going to look like, but it is now on hold. The FDA has held it up and said, ain't going to happen. And the reason it's not going to happen is because of this issue, the tumorigenesis. And the FDA wanted more assurances from the company that these cells won't form tumors. Okay, let's do a little bit of history of human stem cell research. So mouse embryonic stem cells were isolated about 20 years ago. They are easy to obtain and widely accessible to researchers. The potential of stem cells are obviously replacing tissues or organs, repairing defective cell types, even delivery of genetic therapies for individuals with some genetic disease. You can use the cell as a way to repair the genetic defect. Delivery of chemotherapeutic agents for people with tumors. And so what do we know now about human embryonic stem cell research? It all began in 1998. That's it. 10 years since the first isolation of the human embryonic stem cell by Jamie Thompson at the University of Wisconsin. In that same year at Johns Hopkins, John Gearhart derived a cousin of the embryonic stem cell called the embryonic germ cell, which are essentially equivalent. That was really day zero for human pluripotent stem cell work. Now, a critical day was August 8th of 2001, when President Bush authorized federal funding only for embryonic stem cell lines derived prior to that date, August 8th of 2001. Now, at that time, it was believed that there were on the order of 60 to 80 embryonic stem cell lines that would be available to researchers, and that would be sufficient. As it turns out, there are 4. And every single one of those 4 has a genetic mutation, a known contaminating infection, or indeed, they're not pluripotent at all. So the ones that we have looked at are incapable of becoming the cell types that we wish them to become and are useless. So if one is to consider going forward with a project using embryonic stem cells, you essentially have to use embryonic stem cells derived after that date by an independent source. And if you do that, you are precluded from using federal funding for that research. So that's the state of it right now. You can use crappy ES cells that don't work and get federal funding for it, or you can go off on your own. Okay, so let's talk about a few more definitions. So people have heard about cloning. And when people hear about cloning, they think about Dolly. Well, that is called reproductive cloning. So that's making a new genetically identical organism. You heard about that earlier. But there is another form of cloning that is called therapeutic cloning, not reproductive cloning. So therapeutic cloning, let me take you through this. There's a patient and you take a skin cell from that patient, and you take an oocyte from somebody else and take out that nucleus and replace it with the patient's nucleus so that it is now genetically identical to this individual. You then make them into embryonic stem cells, turn them into the cell type of interest, depending on what disease they've got, if they've got pancreatic disease or anemia or had a heart attack. But now you've got a supply of these cells that could repair that person's own tissue and is genetically identical to that patient. So you would not need immunosuppression. You can then transplant those cells back into that individual. So the idea of designer stem cells individualized for a particular patient is what really has driven this concept of therapeutic cloning. We're not there yet. It's 10 or 15 years away, this particular concept. And the reason is the FDA cannot approve every single line for every single patient. There's got to be a master stem cell for a particular application as it stands currently. Now, the other concept you need to know about is induced pluripotent stem cells. This got a whole lot of attention late last year and was really viewed as the way to get around the ethical controversy around embryonic stem cell work. And here's how it goes. You take a human adult fibroblast and you throw in essentially some proteins or genes that basically take that stem cell all the way back in a de-differentiated state so that it is now a stem cell, and now you can conceivably push them toward any particular cell type. Induced pluripotent stem cell. And no ethical issues here, right? Because it just comes from a patient. So the published report said, look, this is the death of the controversy. You don't need embryonic stem cells anymore. And it is very exciting that you can take a fully differentiated skin cell, for example, and make it pluripotent, but there are big-time problems. How do you make it pluripotent? You give cancer genes. You got problems, right? Because now what happens when I try to make a particular cell type for motor neurons or oligodendrocytes, those genes are still there. The cancer potential still exists. There's one more problem. Once you've got a skin cell, it has already gone through thousands and thousands of cell divisions. It's tired. It doesn't want to go back and be a stem cell again, right? And so I don't know if anybody remembers the story of Dolly the Sheep, but Dolly the Sheep was made essentially from a skin cell, and Dolly got tumors at a very early age. She had arthritis at a year of age. She had fatty tumors, and she ultimately had to be euthanized because of the early senescence. She aged too quickly. Same problem is going to exist here. So I think keep an eye out for this. It was very exciting work, but it too is 10 to 15 years down the road. Whereas when you talk about embryonic stem cells themselves and clinical therapies for diseases, that time is now, not 10 to 15 years down the road. Okay. So now I'm going to shift to two applications that we have been taking at Johns Hopkins. And the first one I'm going to tell you about are glial-restricted precursors for demyelinating disorders, focusing on transverse myelitis. So if you imagine the embryonic stem cell as the mother of all stem cells, that makes it both powerful but also a little bit scary. You can imagine that further down the differentiation tree, there are cells that have more limited potential but are not tumorigenic and can't form inappropriate tissues, like cartilage cells within the nervous system. Well, a glial-restricted precursor is just such an example. It is a stem cell that can be isolated from the adult nervous system. We have glial-restricted precursors in our brains and in our spinal cords right now, and they can only turn into two cell types, astrocytes and oligodendrocytes. That's it, nothing else. But if your disorder is one in which there is damage to astrocytes and oligodendrocytes, heck, maybe that's perfect. So we have spent an awful lot of work trying to understand these cells. They were first isolated by a colleague of mine, Steve Goldman and Mahendra Rao, and we have formed a team to further understand them as we've gone forward. And this is actually work done by Steve Goldman, and it is really astounding work that transplanted these glial-restricted precursors into a mouse model of fatal demyelination. This is a mouse called a shiverer mouse, and the mouse shivers because it doesn't have any myelin. And the mice ultimately shivers itself to death because it doesn't have any myelin. So Steve Goldman took these cells and transplanted them into the shiverer mouse. And this is looking under a microscope. And what you're seeing is broad areas of the nervous system now showing myelin. The myelin is derived from the transplanted cell, not from the host. Here's a higher power view, which is what we do. We look under the microscope, and you can see that there's lots of these green myelin sheaths that are wrapping themselves around the neurons. And guess what? The mouse stops shivering. And what Steve, this was from 2004, but Steve just published a paper a couple weeks ago in which these mice not only stop shivering, but they're cured. So instead of dying at 30 or 40 days of life, they live as long as he's followed them out to 1.5 years. Amazing stuff. But it really points to the potential of what these cells, these GRPs, can do. And if you look under higher power, including electron microscopy, what you're looking at here is myelin sheaths wrapping around an axon here. And this is what myelin should look like, because that's what oligodendrocytes making myelin do. They wrap their arms around this axon time and time and time again, and that's what you're seeing here. And so this just proves that the myelin that they form is good stuff, really good myelin. So we said, all right, well, that's cool, but we don't work in shiverer mice, and that's a genetic pediatric disorder. What we really wanted to do is to study it in transverse myelitis. Well, there was no animal model of transverse myelitis, so we went about and made one. And what we did is we immunized animals with a myelin protein, and I won't get into this, but we gave them cytokines, which you've heard about now. You're all well-versed in cytokines here, into the spinal cord, including one that you've heard a lot about, which is interleukin-6. And what happens? The animals get sick. They get inflammatory demyelination in their spinal cord. And so they look very much like transverse myelitis patients. Why did we do this? Because we wanted the first indication for these cells to be in patients with transverse myelitis. We then transplanted them with cells that are called Q cells, which means they are human glial-restricted precursors. This is another microscopic image, so for those of you who don't look at this very commonly, don't worry, except it shows a spinal cord of a rat which looks identical to the spinal cord of a patient with transverse myelitis. There is an awful lot of inflammation here. This is the event that is the initiating event in human transverse myelitis. And when we look more carefully, in fact, these rats have all the hallmarks of true transverse myelitis. This is the back part of the spinal cord that mediates sensation in this rat. It should be blue, but it's not blue. It's completely demyelinated, as opposed to, say, this area, which is nice and pretty blue, and that's an area further up which is unaffected. So great. So we've essentially made a rat model of transverse myelitis, which allows us to explore the biology of these cells now transplanted into that lesion. And the question is, can we repair that area to make the rat better? Because that's what the FDA wants if we can make the human better with transverse myelitis. So I'll just show you a few pretty pictures. This is just one example of now we've transplanted the cells into the spinal cord. We've waited. We've allowed the animals to survive. We've seen how they've done. And now you can start to do pretty stains looking at what they become. And this is what I think is a particularly pretty image showing the green fluorescence here. We use a jellyfish protein to tag our cells. And so every time we transplant one of these GRPs, we make them fluoresce like a jellyfish so we can see them under the microscope. And so here you see the green fluorescent jellyfish cells that indicates that the transplanted cells have survived. And then we can ask what they have become. In this case, they have become astrocytes, the glial cells that are the support cells within the spinal cord that become damaged in transverse myelitis. Now, there are other cool ways, and these are just some cool tools that I'm going to show you about, that we can identify what they are doing. Because it's very important for us to take this rat and we let it live for a long period of time, but we need to see how these cells are doing. So one of the tricks that we've developed is we throw in a little bit of iron into the cells just prior to transplantation, because if the cells have a little bit of iron in them, you can now do an MRI, and I can identify precisely where the cell is over time, over months and months and months in that animal. And I'll just show you the summary of this. This is basically taking MRI sections up and down this rat's spinal cord. We've reconstructed the bones of the rat here from the MRI, but what we're really interested in is what happened to the cells. And it turns out they migrated up and down the spinal cord, they survived, they became astrocytes, and they became oligodendrocytes. So we could define that simply by giving the rat an MRI every 4 to 6 weeks to find out how far they went and what they actually became. This is just another example, a microscope image this time, in which we're again looking at the spinal cord, and each of these blue dots refers to one of these cells that has survived and has become the cell type that we're interested in, oligodendrocytes or astrocytes. Another example of this, we can see these cells initially within the spinal cord stay right where we put them, but by 10 days later are starting to migrate their way up and down the spinal cord, seeking out an area of inflammatory demyelination. So that's beautiful, because if we had to put them throughout the entire lesion, that would worry me. So it turns out we don't have to. They will seek out those areas of inflammation and demyelination. We don't have to worry too much about it. Now, I won't go into this, except to say that I don't know who of you has ever had an evoked potential, whether it's a motor evoked potential, a somatosensory evoked potential, or a visual evoked potential. It's a way electrically to ask how the nervous system is working. Okay? So we said, well, we'd like to do this in our clinical trial, and so the FDA said, fine, do it in your rats. Which isn't that easy here. In a human, I get it. We can do that. But to do this in a rat is pretty hard because they're so small. So it took us about 4 years, actually, to figure out how to electrically hook up the rat so we could figure out conduction across the spinal cord. And although this doesn't mean much to you, it means an awful lot to me. That's why I had to show it. Because what happens here is we're detecting transverse myelitis electrically in these animals. What you can see is that over time, just like happens in humans, the electrical impulses fade and go to zero. And that's what we need, because if we're really going to prove that they work, we can't ask the rat how it's feeling. We need some electrical measure or some imaging measure. That's what the FDA says we need to have. And each of these lines here then is an individual rat where we've looked at these particular measures, and I won't go into this except to say that guess what? These electrical signals go back to or near normal after the injury if we've transplanted these glial-restricted precursors. Now that's early. We just got this data actually last week. But it's very exciting to say that here we've got now an objective measure saying that the cells are doing what we hoped they would do and what we thought they would do, and we can define that. And that's what the FDA has told us we need. And so hopefully that will be sufficient as we get that all together and submit it to them by the end of this year. Okay. A few more, and this just shows that when we look under the microscope, they actually become myelinating cells. Red here indicates the human glial-restricted precursors, and the yellow and green here indicates myelin. So that's cool, because we know this is demyelinating, and when they start to express myelin, that's good, because that says that they are remyelinating area. So, why transverse myelitis? Well, you know why I would want transverse myelitis, but we had to convince the company, Q Therapeutics. We also had to convince the FDA that this would be the appropriate target, because this will be the first nervous system stem cell clinical trial. This is going to be a big deal. So the real reason that we had to convince them is, look, you've got one lesion in one small area, a lot of disability, and all of that disability is due to that one lesion. It's much more complicated if you got 20 lesions and you're asking me to repair 20 lesions. But if I can make the argument that if we repaired one lesion, you could have a very significant functional benefit, that would be very important. And so it was on that basis that both the FDA and this company elected to go forward. It will be a single-center clinical trial at Johns Hopkins. It will involve acute to subacute transverse myelitis patients. The first trial really is designed for safety, so we don't know exactly what it's going to do, but it is a safety trial, and it will involve patients who are 6 weeks to 1 year out from the acute transverse myelitis episode. Many of you will say, that's too bad because I'm outside of that window. But remember, we have to get this right up front. We have to pick the best patient cohort in which to test this concept. If we are successful, then you expand out into other cohorts. But you've got to get it right early, or this thing will not survive to get to those second and third generation clinical trials. So what I would say to you is, remember, the first trial is a safety trial. You really don't want to be a part of it unless you have to. Let us figure out the details in the new onset of transverse myelitis patients, figure out how safe these cells are, but get it right, and then move on to the second and third generation trials. Okay, last few slides. I'll summarize it very quickly. Back in 2006, my group showed that we could essentially rewire part of the nervous system in rats. This has now been replicated by 3 other groups who did the same thing. Essentially, in models of paralysis, you can use stem cells to rewire this part, resulting in functional recovery. So that is now routinely accepted as being possible in rats. There's a group in Italy, in Milan, that has done this, and that paper is coming out in a couple weeks. There is a group at UT Galveston that has done it, and there's a group at UC Irvine who hasn't published it yet, but it's the 4th time that this has now been done, starting with ours. So I've shown this group these old studies now, because they're now several years old, but it's just to make a point that we can make motor neurons in the spinal cord. Here we've got the jellyfish green again, right? But now we're looking at motor neurons, not oligodendrocytes. The target is different. It's not demyelinated lesions. It's paralysis from motor neuron injury. We got these motor neurons to extend their axons out of the spinal cord and to form neuromuscular junctions, which is the specialized synapses with muscle, resulting in the rats getting better again. And that was the point that has now been replicated by several other groups. Was very exciting, the idea that you could recreate a functional part of the nervous system after injury. So this data now is not mine. This is data from Hans Kierstead, my collaborator, who has said, well, that was very cool what you did, but I want to do this with human embryonic stem cells. What I did it with was mouse embryonic stem cells, and he's generated literally billions and billions of these cells. These are human embryonic stem cell derived motor neuron progenitors. And they grow very rapidly, they are very pure, they are very safe, they are non-sanctioned, non-federally approved embryonic stem cells. And this just shows what they do. So, what are we going to do? We hope to take this to the FDA for a clinical trial, meaning we have to have proof of concept. And you'll hear about this as you get more stem cells going through. But one of the things they want to know is proof of concept. Got to be efficacy. And we are in the midst of that. But essentially, we are almost through this and already know that the human embryonic stem cell-derived motor neurons do exactly what I had shown with the mouse embryonic stem cell-derived motor neurons. They're behaving nicely. So we believe that we're done and have solid proof of concept. That's good, but the FDA wants more than that. The FDA wants preclinical safety. And that's actually what is most important to the FDA, specifically tumorigenicity, toxicity, and biodistribution. Tumorigenicity, simple. Do they form tumors? Toxicity, simple. Are other tissues or organs injured by the cells? And biodistribution is where do your cells go? So we are now in the midst of a preclinical safety study that is about 4 months long of a minimum 6 months. So we'll start to gather this data in the fall. Don't know what it's going to show. If there are tumors, then it will obviously be a break. But we believed, actually, that we would see them if they were to exist. In the first 3 months, none has been seen. So spinal muscular atrophy is a 100% fatal genetic disorder. Babies are born floppy. They can't suck, they can't feed, and they die. There's no treatment. There's nothing that slows the progression. These babies are diagnosed in infancy, and for many reasons, this is the perfect disease in which to test the motor neurons. They die exclusively because the babies don't have the motor neurons. Myelin is a complicating factor which isn't there in the babies. We know that the axon guidance cues, basically the road signs telling them where to go, are there in the babies and not necessarily in adults, and the distances are short. The point of that is, why would we do that over transverse myelitis? Because we don't know this stuff yet. So do it in the babies first, get it to work, and then we can start to sort it out and think about other patient groups in whom to test it in, including transverse myelitis. So the proposed clinical trial, human embryonic stem cell-derived motor neuron progenitors, high purity, highly differentiated population of spinal cord motor neuron progenitor cells derived from human ES cells. They are currently being manufactured under good manufacturing practices, good tissue practices, and aseptic processing. This is being driven by a company in California, which is called California Stem Cells, Inc. The primary and secondary objectives of this study. The primary objective is to evaluate the safety and tolerability of escalating doses of motor neuron progenitors into the spinal cord. The secondary objective is to evaluate the potential trend towards efficacy or effectiveness of these motor neuron progenitors. We hope to actually do this transcutaneously, which we also hope to do in the transverse myelitis study, which means a needle through the skin into the spinal cord, rather than a spinal cord surgery and laminectomy. We are talking with the FDA about that because it's quite simple if you do it that way. There's no sutures, there's no nothing. Now the FDA says, yeah, but you can't see it as well if you're going through the skin than if you open up the spinal cord. So these are issues that we have not yet decided. I see it both ways, but I think the easier way to go is what we call transcutaneous through the skin. And we will just give 4 injections into the babies with spinal muscular atrophy. We, of course, are looking at safety, but we are looking at a series of surrogate markers to see evidence that they are working, and we will look at how the babies are breathing. We will look at how big their muscles are. We will look at their x-rays and see how they're doing, and of course we'll look at survival. And so I think that is it. Those are my 2 daughters. We all show our daughters here as we end our talks. And questions? Yeah. What? Boys? I don't know. It's something in the water.
Two questions. One, maybe overly simplistic, but embryonic stem cells is such a loaded term. Why hasn't the scientific community adopted the blastopods?
The question is, why haven't we changed the terminology? Because it's such a loaded term. I think it's too late. I wish it weren't so. I wish we could. Blastocyst-derived cells. You would have never heard about them except from me at a lecture rather than on the nightly news. I think it's probably too late. I think there would be a real uproar if we decided to rename them at this point. But we should have at the time. Ooh, wasn't me, though. Yeah. They obtained their stem cells from a donor who gave consent, had her health checked, had a whole series of tests both to her and also to the father and to the embryonic stem cells derived. It was done with independent funding provided by the company. No, so the question is, are they adult stem cells? No, they are derived from an in vitro fertilization. To get motor neurons, which was the last part of that talk, the only stem cell that can make a motor neuron is an embryonic stem cell, period. Now, to make myelinating cells, you can get that from other things. But to make motor neurons, there's only one source, and it's the embryonic stem cell. Is there the same problem with embryonic stem cells worldwide? No. Virtually every country in the world is further advanced in their acceptance of embryonic stem cell. This includes Iran. This includes China. This includes Israel. It includes every European country. We are unique in our resistance to this. The question is, are there human studies going on elsewhere? I wouldn't call them studies, really, because the problem is, although they have more tolerant acceptance of embryonic stem cell, they don't have the biological underpinnings and the clinical infrastructure to do them. So there's stuff out there, but it's not done very well. And in fact, some of these cells, some of these studies are dangerous. You can go get stem cells in China, Uzbekistan, Tijuana, Trinidad, Tobago. Chitra will probably hook up something in Trinidad and Tobago. You can get stem cells at a variety of places, but they're not regulated very well and they're dangerous. Some of my patients have died.
Yeah, switching from embryonic to glial-restricted precursors, the trial you're talking about with TM, you're hoping to enroll next year, that's the GRP.
Right. The TM trial next year will be GRPs, glial-restricted precursors.
Whereas you say for some of the stem cells are trying to get the sort of motor neuroregeneration, but that doesn't have schedule.
2009.
So that might happen in 2009.
What we need to do, it depends. So the question is, what's the timetable for the motor neuron trial? And it depends what the FDA says, but we're going to ask them for permission to do this in late 2008, early 2009. Now, they may say no, and that would be very appropriate. It depends upon their level of comfort with the data we've got. Again, the embryonic stem cell is a little scarier, so I think the bar needs to be higher for that. Conversely, what the FDA says is there is another difference here. Transverse myelitis is bad, but it's not fatal. Spinal muscular atrophy is fatal. And so the bar can be lowered a little bit for a disease which is otherwise 100% fatal with no treatments. So I think it's conceivable that both will begin in 2009. I know the FDA will be asked to give us permission for both for 2009.
So when you mentioned your colleague, I didn't catch his name.
Hans Kierstead. No, he's at University of California, Irvine. That's Hans Kierstead.
So that's where this would happen if it happened.
No, it would happen at Hopkins. The way this is going to work is these cells are going to make transcontinental flights. In other words, the cells are going to take the red eye, are going to arrive at Hopkins, and be transplanted at Hopkins into the babies. They don't have a clinical group there. The UC Irvine group does the manufacturing and some of the preclinical toxicology, biodistribution, that kind of stuff. They do not have a clinical setup for actually transplanting the patients. Yeah?
How does someone get on Hopkins?
How do you get to be recruited for a clinical trial? This is not a trivial question because the FDA views this as a very important issue so that there's not kind of nepotism or favoritism in who gets in clinical trials. And the FDA has told us that they will control it, that the individual sites, especially for stem cells are so loaded and there's so much passion behind them that this is going to be scrutinized very heavily. And the FDA wants to control how people are given entry into the study. Now, for the transverse myelitis trial, it's going to be acute patients. So it's going to be patients who are within a time frame. And not too many people fit that.
What about the
The cytoxan. For the cyclophosphamide study, that's, completely different. But that will be a phase 3 clinical trial. That will be at multiple sites throughout the United States, 70 patients per arm, and anybody who's interested will be given access into that if they meet the criteria.
let's say Hopkins is one of the sites. How do you let Hopkins know when you're ready?
You'll hear about it. There will be a lot of publicity about this as it gets going. The National MS Society will list this trial once it's up and running, and the NIH, since it will be a registered clinical trial, will be on the NIH website. At that point, the contact numbers for any of those trials, whether it's the cyclophosphamide or the stem cell trials, will be there. You can contact the clinical coordinator and get evaluated. Sir? Say that again? Yeah. Do I follow them? Yeah, so we follow them. We're not sure which of the two cell types, whether it's oligodendrocytes or astrocytes, are more critical for the recovery, but you get both, and either or both may be important in the recovery. Yeah. Does banking of cord blood fall into this? Specifically into this issue, no. But I get that question a lot. I don't know what to say. As of now, cord blood cannot become relevant neural cell types. There are two, so that's one of the two problems with cord blood. The second thing is, as of now, cord blood cannot be expanded. So it is what it is. You can't make more, you can't keep growing it. But that may change. And so I don't know the right thing for that. There are literally hundreds and thousands of cord blood registries available. If those cells ever do become valuable, it costs, $1,200 plus a monthly fee to keep it. So I think given what we know now, some people might wish to do it, but it doesn't make sense for everybody. Yes. Yes, ma'am. Will a new political environment be an asset? Heck yeah.
We're questioning and answering, so we're getting more praise.
Okay. Yeah. Yeah, so there is the SMA Treatment Acceleration Research Act, which is now making its way through the Senate, I'm in favor of it. I think if we can start to really effectively treat any of these diseases, it will have a spillover effect to the others. So I'm supporting that one. I've supported the expansion for federal funding for embryonic stem cells, which went before President Bush twice, and 2 of his, I think, 3 vetoes in his 7 years have been to veto that. There's no sense in that coming up again. It's not a 2/3 majority. You can't override the veto. But that will be, I believe, the second bill passed in January with the new administration.